Hi, everyone. It's Dr. Linder. In this short video, we're going to be talking about the neuron. As a quick review, when we discuss neurology, remember there's different types of cells that are involved with the nervous system. Typically, I like the word neural, calling it the neural system instead of nervous system. We have the neuron, which is the only electrically excitable cell. And then we have neuroglial cells, and you'll be learning a little bit more about them in your lecture portion. Things like Schwann cells, oligodendrocytes, satellite cells, microglial cells, ependymal cells, astrocytes. Those are neuroglial cells or neuroglial cells. We're going to be focusing on the neuron. When we look at the neuron here in this picture, we'll see that we have a few main parts that stick out to us. And I'm going to say three main parts, and then we'll break them down into smaller bits. But you see these branches here on the outside, those are dendrites. And then we have the soma, which is the cell body. Soma, or cell body. And then we have this structure called the axon. And at the very, very end of the axon, we have the axon terminal. Okay, and at the end of the axon terminal, where we have this little box, this little square box right here, this is called the synaptic end bulb. The synaptic end bulb. And if we think back to myology, when we did muscular physiology, what is stored in that end bulb are those synaptic vesicles that have neurotransmitters in it. And at the neuromuscular junction, that neurotransmitter was acetylcholine. And you see the arrow moving in this direction. This arrow indicates the direction of the nerve impulse. So it goes this way. This dotted section that's outlined here, this dilated part of the axon, they call this the Axon Hillic. The Axon Hillic. So now here, what we're looking at is part of the neuron. And let me just show you a few other slides of that. Here, right? So we're going to focus on the gray one, not so much the yellow model. And what we're looking at is this portion here. This portion here is this. And then this portion here is this, right? So you can see that right here, they cut it. And this slice would be right here, right? So you would take this end here at the bottom and glue it right here. But for the size of this model, when we do it actually in the physical lab, it's just too long. So they had to kind of slice it into two parts. But understand that this portion, right, these are the dendrites up here, here's the cell body, and here's the axon. This axon would continue right here. We would glue this right to the tip. Okay, so key parts. We have the dendrites here. Dendra means branches in Greek. So those are dendra. The soma is this entire structure, which is the cell body. It's another way of saying cell body is soma. In the center is the nucleus. And here is the axon. The dilated portion here at the beginning is the axon hillic. And between the axon hillic and the axon is a specific region called the initial segment. Sometimes they call the initial segment the trigger zone. the trigger zone. Okay, let's move on to the next slide. And we can see that now we're looking at the axon region, which is this entire structure here. Okay, so this is the axon. This is still the axon. Here in the center, they call this the axoplasm. The axoplasm. This structure here on the outside, whether it's here or here, this is an entire Schwann cell. From here to here, this is one Schwann cell. And what the Schwann cell does, let's say this is my pen right here, is the axon. This is a Schwann cell that's going to wrap around it, okay, like that. And it's going to create this kind of like jelly-like ring and insulation wrapping around and around and around and around and around and around the axon. And what it creates as it starts to wrap around the axon is this protection. The protection is what is referred to as the myelin sheath. The myelin is made up of fat, lipids, cholesterol. It's really a fatty insulation. Okay, and this is why we need cholesterol in the body, right? When people take cholesterol medication, many of them get what's called neuralgias or paresthesia, which is neurological symptoms due to the lack of insulation and protection that the body can make wrapping around your neurology. Okay, so you have to be really careful with that. Paresthesia is a term that we use to describe pins and needles sensation of the fingers or the feet. They call it paresthesia. Now, between one Schwann cell and another Schwann cell is this region right here, which has an arrow here. This is if it's sliced. This little invagination or indentation is the same thing as this right here. This indentation here or this indentation here is called the node of Ranvier. The node of Ranvier. And many textbooks say it's a region that's unmyelinated. It's a very, very thin junction region that's unmyelinated. And as the action potential is traveling down the axon, they say that it leaps from node to node, right? It leaps over that unmyelinated section. That's the action potential or the depolarization. Okay, and that's what they refer to when you get to lecture. You will learn about saltatory conduction. And saltatory conduction is the method of which the action potential jumps or leaps over each node of Ranvier. In fact, the word saltatory comes from the word saltare, which means to leap or to jump. Okay, and right here is just the nucleus of a Schwann cell. Okay, there are some other structures, but if I don't mention it, I'm not going to hold you accountable for it. Let's just, you know what, for funsies, let's just look at the yellow one. And again, just to show you on another model, some of the same structures that we covered. So the dendrites are these yellow branches. The soma is the cell body. Right in the center is the nucleus. Here is the axon, right in the center. And the beginning of it right there is what we call the Axon Hillic. And again, between the Axon Hillic and the axon, kind of still in that beginning stage, is what we call the initial segment. Sometimes they call it the trigger zone. What lines, or what is superficial to the axon, is the myelin sheath. So this is the insulation or myelin sheath. You can still refer to it as the Schwann cell. They're both acceptable. If you said Schwann cell or myelin sheath, you would get credit for either of them. And here's just a close-up view. We see the dendrites. We see the cell body or the soma. Here's the nucleus, the Axon Hillic, initial segment, axon, the beginning of the Schwann cell or myelin sheath, Schwann cell or myelin sheath. This is just the nucleus of a Schwann cell. And again, where one Schwann cell and another Schwann cell unite, where they come together, that's the unmyelinated portion referred to as the node of Ranvier. That's where saltatory conduction takes place, or the action potential leaps and jumps over that unmyelinated portion. Let's see what else we have here. Right here, this portion of the neuron is called an endoneurium. Remember, there was a endomysium, there was a epimysium and a perimysium. We have similar layers when we talk about the neurology, where we have an endoneurium, we have a perineurium, and we have an epineurium. This right here is the endoneurium. These right here, I know they have a bunch of different names for them, but these are like right here, synaptic end bulbs, right here, synaptic end bulbs. That's all they are. You could see it as a close-up. Probably have some neurotransmitters in there.